Good morning, everyone. I'm Justice, and I'm happy to welcome you to Pediatric Grand Rounds this morning. Could I have the first slide? I'd like to begin by acknowledging the land on which Sick Kids operates. For thousands of years, it has been the traditional land of the Huron-Wendat and Petun First Nations, the Seneca, and most recently, the Mississaugas of the Credit River. Today, Toronto is home to Indigenous peoples from across Turtle Island. Sick Kids is committed to working towards new relationships that include First Nations, Inuit, and Métis peoples, and is grateful for the opportunity to share this land in caring for children and their families. So next slide. Today, I'm very happy to introduce the Jacob's Ladder Norman Saunders International Research Prize, which is an endowed lectureship given by the Schwartz family in honor of their son, Jacob, who suffered from Canavan disease. The Schwartz family promoted genetic screening for rare genetic diseases in Canada, as well as research and go back one slide. Oh, I guess you're fine, never mind. Rare genetic diseases in Canada, as well as research and development for the diagnosis, treatment, and potential cures for neurodegenerative conditions through their foundation. So I'd like to remind you that the prize ceremony will occur after the question and answer session of the lecture using this same link. And this will be followed by an unplugged session for trainees and others who are more interested in personal information about Dr. Brais. This requires a separate link. Next slide. I'd like to remind you that questions will be addressed at the end of the presentation. So if you'd like to ask a question, please use the Q&A function on your Zoom. And now the next slide. So I'm very happy this year to present the prize to Dr. Bernard Brais. He is a neurogeneticist and historian of medicine, a professor of neurology and genetics at McGill University. He's also co-director of the Rare Neurological Diseases Group of the Montreal Neurological Institute. I find it extremely important that Bernard is winning this. The prize is named for Jacob's pediatrician, Dr. Norman Saunders, and two of Bernard's mentors, Dr. Charles Schreiber and Dr. Eric Showbridge, have won this prize in the past. So Dr. Brais, in following on this wonderful tradition in Canada, has played a huge role in diagnosing rare neurodegenerative conditions, as well as using genetic knowledge to inform treatment options. So without further ado, I'd like to turn this over to Dr. Brais, who will give us his presentation.
Good morning, everyone from Montreal. Sadly, I cannot be with you this morning. I want to thank Ellen and Jeff Schwartz for awarding me this very prestigious award. Thank you very much. Thank you to your foundation for all the work it has done over the years, and thank you for Monica and the committee for selecting me. I think we're going to talk about tradition today in the sense that I'm going to discuss ataxia in Quebec, and Quebec has a special genetic history, and we will dwell on that quite a bit. And hopefully a few of the lessons I've learned will be shared with everyone and make you understand that to treat rare disease, it needs a lot of people. It needs a lot of transmission of knowledge. It needs a lot of solidarity. And hopefully many of the stories I'm going to tell you today will contribute to that. So I have no disclosures. So I'm very happy and honored to receive this prize because as Monica stated a few minutes ago, two of my mentors received it in the past, and so it is tradition, a first mention of tradition in this talk. I need to say a few things because I think it will shed some light on my original professional course. I decided to become a neurogeneticist when I was 22 years old, so we're talking about in 1982. And the reason was Charles Schreiber, who accepted to see a young, over-enthusiastic French Canadian who was fascinated by history, fascinated by medicine, and who didn't know how to bring all of that together. At the time, I was already interested in neurology, and Charles just took the time to say, look, if you really want to do history, do applied history, do history of the genetic diseases of Quebec. And that makes sense if you're interested in neurology, because 50% of hereditary disease have neurological manifestations. There was one caveat. He said, if you really want to do all of that, be a historian, be a neurologist, be a geneticist, you'll have to complete the training in the 3 disciplines. For the younger people listening today, I'm not sure it was a good idea, but still, enthusiastic and young as I was at the time, I decided to do that. And that brought me to 3 years of very fascinating work in France and London and Paris, in fact, to study this man, Jean-Martin Charcot, the father of modern neurology. And though I wrote essentially on social history of Jean-Martin Charcot, it allowed me to enter his mind, enter the mind of the clinician who was really into discovery. At the time, it was discovery of disease by comparing clinical presentations to pathological. It was his famous anatomopathological method. But it was based a lot on observation. Jean-Martin was just a fantastic observer. So when I came back to my residency, then I knew I wanted to be a researcher, but had not been exposed to laboratory research. It was an amazing time. It was the first gene, it was Duchenne gene. It was just a time of great excitement. And I decided on the recommendation of George Carpati to do almost a year of my residency working with Eric Schubridge, who, again, for the young people listening today, it was a time of the beginning of PCR. So the world was changing, the neurogenetic revolution and the genetic revolution was happening. And I was there to surf that huge wave. And I'm going to share with you some of the excitement, some of the ways we've surfed, not only for the pleasure, but always to help patients. So again, the method I've used over and over, in a sense, the talk I'm giving today is about applied history when you're treating a population or members of a population, was derived to, in some extent, from this observational obsession of Charcot. So the method is essentially, this is a very classical picture. It's a picture of the first Charcot-Marie tooth case that Charcot described. And he used a very French descriptive term to describe her legs. He called it inverted champagne bottles. You're going to say it's very ethnic. But that image has stayed and we still use it in our teaching. So if you see one case of it, and then you see 2 cases of it, in Quebec that is, then you can always look at the experience of other physicians, neurologists, people who have looked at and worked in that population over the years. And often you will find old publications in the 20s, 30s, 40s of people describing similar cases. It's the accident of having a shared gene pool. You will have people with the same disease in some region of the province. So I then, and I must say I'm obsessed about geography, I would then map the patients to the area. So often when I see patients in clinic, the first question I ask is, where do they come from? Where do their grandparents come from? And that already sets the stage for me to place them on the map and therefore influence my final diagnosis. And then obviously collecting families and really ensuring that it looks like and like a genetic disease. So the objective of the talk today is in fact to review an entire tradition, the study of ataxia in Quebec, obviously very in an accelerated mode, but I think there's a few lessons there to be learned. Then discuss some of the conclusions as we see them now about the impact of diversity and some degree of the variability of phenotype in Quebec. And then use a story which I think probably promoted the people of the committee to award me this award, is the idea that my work and contribution to a group who's working on autosomal recessive spastic ataxia of Charlevoix-Saguenay, a flagship rare disease, neurodegenerative rare disease in Quebec. So for the ones who are less familiar about Quebec, Quebec is huge, but most of its population are in the, this is the St. Lawrence River, or around the St. Lawrence River, with the Quebec and Montreal as being the 2 larger cities, but often a very important region, the region I come from. And that's not why it's important. Saguenay here, Saguenay-Lac-Saint-Jean, which you'll hear about at different times in this talk. So these are the 26 touristic regions, But in fact, they to some extent overlap biological regions, because those regions were settled by either rivers, and then with time, migrants from the riverside moved into the land. And so a lot of the people in every of those regions have shared genotypes. So in a sense, they are more likely to share recessive mutations. So the story of ataxia starts with this man to a large extent. And the story of neurogenetics in the world starts from this man, a French-Canadian, brilliant guy called André Barbeau, who died unfortunately too young, who was active in the founding of neurogenetics as a specialty, was responsible for the first, but mostly the second Congress of Neurogenetics held in '67 in Montreal, and was known all over the world. And one of the initial contribution was to put Quebec on the map as the place where there was a founder effect for a disease called oculopharyngeal muscular dystrophy. And I'll talk a bit more in a second about it. But Barbeau did something very interesting, is that he realized that this disease was not distributed equally across the territory. And in fact, there was more of this disease in a region called Montmagny-Lillet, north and on the south shore of Quebec City, and tried to do a lot of genealogical work, did a lot of archival work, in fact, left tremendous archives about that to one of his spiritual students, Jean-Pierre Bouchard, in fact, gave them to me a few years ago. So we did a lot of work on that, but I think it illustrates the methodology, the description of the first really good description of the case with a French-Canadian family living in the Boston area. Then a very good description in the 50s by a tremendous neuropsychiatrist on the French side, Romain Agnew. And then the description in the New England Journal of Medicine of the Jewish family of the disease. And then Barbeau, who at the time was bedridden, read this paper and said, wait a minute, I've seen this. And then he started in the late 60s. He spent, in fact, 2 summers in the region where this was more prevalent. And then saw families over the years, and then transmitted these family trees to Jean-Pierre Bouchard, who then joined forces with Guy Rouleau, my mentor, and put me on a golden platter, this project as my PhD project, to find the mutation for this really flagship autosomal dominant French-Canadian founder effect disease. So I won't talk about OPMD, I'll talk a bit more about it tomorrow for the ones who are attending. So let's go back to Barbeau. He did something also that's, I think, is really pertinent, thinking of the Jacob's Ladder Foundation also, is that he got very close to a fellow with Friedreich's ataxia. His name was Claude Saint-Jean, brilliant, publicity-oriented person who developed a foundation, really a star of what it is to live with a rare disease in Quebec. Created a foundation that has moved over the time and now is called Ataxia Canada. And with him raised a lot of money. And that money was used largely to do research. And I'm not talking about small scale research. In fact, it developed a consortium, and that consortium was international, led by André Barbeau and to a large extent also Jean-Pierre Bouchard, and it published over a little bit less than a year, 10 years, 149 articles. It created the first criteria for Friedreich's ataxia. It put Montreal and Quebec in general on the map for ataxia research. And it prompted the work of his spiritual descendant, Jean-Pierre Bouchard, who, coming from the wonderful, and for you who want to visit Quebec, I would highly recommend the Charlevoix area. So this is the Saguenay I mentioned earlier. This is the Charlevoix, a gorgeous, really one of the most beautiful regions in Quebec. And he was born in Baie-Saint-Paul, and he described a disease which was regional. Think of OPMD as a kind of regional disease. That same obsession, Bouchard described autosomal recessive spastic ataxia of Charlevoix and Saguenay. Saguenay is an offshoot population-wise of Charlevoix in many ways. And so that put on the map a new founder ataxia in French Canada. And for decades, it was thought that RSACS was a unique French-Canadian disease, that kind of odd population of Eastern Canada that was different. I can tell you now, it's the second most common recessive ataxia in the world. So what we find in Quebec is relevant for the world, and I think Jean-Pierre Bouchard showed that, and I'll mention it in a few minutes again. And I'll come back to Ataxia a little bit later for the second half of the talk. Then I came on stage really for the Ataxia in 2002 when I took over Brais Clinic. So it was 30 years of history of following between 400 and 600 Ataxia patients. So I took over from Massimo Pandolfo and from Luc Marchand. And then one of the first clinics, I was struck because I saw a Charcot patient. I saw a patient who had Ataxia, but had also the same features of atrophied feet and hands, almost skeleton-like hands. And it was like reliving the Charcot experience. And I asked around, and I remember at the time, Luc Marchand, who had followed the clinic, said, I'm not sure I saw any other cases of that. And then the following clinic, he said, look, Bernard, I have 2 in my office. So rapidly, over a few months, we decided to collect all those cases. And then we rapidly realized that though there are some all over Quebec, most of them are in the northern peninsula of Gaspésie. And in fact, these patients are all homozygous for the same mutation. We were scooped for the gene, but still, and this research prompted the interest in Ataxia, and the first author there, Antoine Duquette, who's now the neurologist taking care of this clinic at the CHUM and very active in Friedreich's research and other Ataxia projects. So that was a kind of other regional founder effect for an Ataxia in Quebec. Then Bouchard struck again. He had seen many cases. Quebec City, he was working out of Quebec City. He's retired now, and seeing a lot of patients from both, which is this on the other side of the St. Lawrence River, and with time collected a large number of patients who, for him and for his student Nicolas Dupré, were more or less pure cerebellar ataxias, adult onset. And they put them all together, and with the help of Guy Rouleau, managed to find the gene and published a mutation. Again, it came out as a regional, because it's associated with the region of Quebec, a kind of regional founder disease in French Canadian. I can tell you now, it's 2 or 3 in frequency in the world. So again, perceived as a French Canadian disease, more common. The phenotype has expanded. Even in Quebec now, we know it's a frequent cause of spastic ataxia, not only of pure cerebellar ataxia. It's a very wide spectrum now of disease, but still, it illustrates that in Quebec, because of more frequent mutations, you could have a kind of a more pure presentation. Pure is maybe not the right word, but I mean a more similar presentation or more homogeneous presentation that allows the clinician to see, as I described earlier, the same repeated image. You can see the 33-year-old and the 42-year-old, different families, but they look the same. They're not of the same family, but they are. And as we will review, they share the same mutation. That's why they present more homogeneously. So that was a major passage. Continuing in the same vein, the same exercise of using founder population, we identify the mutations in a new gene for a rare form of ataxia, posterior column ataxia, from a small village called Pespébiac. In fact, very beautiful place, and a very interesting population history. It's a place that was founded by Basque fishermen. So it has a very unique genetic background. So in fact, we were able to describe one of the largest families of this rare condition. So we had another northern eastern founder effect for an ataxia. Then, because of my interest in ataxia, I was approached by again parents who wanted me to do more research on the disease of their child. And the child had leukodystrophy, hypomyelinating leukodystrophy. And I was fortunate at the time that I had a very, very bright student, Geneviève Bernard, who was doing her fellowship, clinical fellowship with me and Guy Rouleau, who decided to take on this project with me. And we realized that, in fact, I had seen other cases, and we managed through their little foundation to recruit other cases, many from the same region. And that allowed us to find the gene for this hypomyelinating leukodystrophy in French Canadians on a small set of families, most people carrying the same mutation. It turns out that it's the second most common in the world hypomyelinating leukodystrophy. And now the work has progressed to an immense degree, and Geneviève has taken the leadership role and ensuring that we continue to strive to find to better understand, to find treatment for them. Why I was always interested was because a lot of white matter disease are associated with cerebellar presentation, and all those patients were ataxic. So that was really my major interest in it. So again, a disease that was in fact on the other side of Quebec City in a small area called Brais. And then there was a large group of patients that we were following over years that had an overlapping phenotype. And it turned out that many of them came from this Portneuf area just south of Quebec. And they had mutation SPG7, which is also now in the top 3 for recessive ataxia, though it was described initially as spastic paraplegia. So if we, going to the second part of this talk, I'm going to try to bring some of the elements to make you understand why is this happening, and also what does it tell us, or how does it help the clinicians in their diagnosis, and also how homogeneity of a phenotype and genotype is now becoming diversity. So if we, in the clinic, if I see an ataxic patient for the first time, I have this kind of map in my mind. And that's why I want to know where they come from. So that can be in part by their name, because there's a geographic distribution of family names in Quebec. So you kind of get an idea, at least where half of their family comes from. And then you then dig in and try to find out where they come from. I must say, and this is in a way a joke, the further south you go, the least the people know where they come from. So the urban reality with time drowns this by mixing between people, but also just because there's none of that physical, what we call belonging or appartenance in French. So people don't really know. So we have to work on them a little bit more. But the people who come from the regions, they really tell us, and then it becomes. So we know where there's the most common autosomal dominant ataxia to date is SCA2, and it's associated with a village in Gaspésie also. And then we know, depending where they come, the Friedreich's ataxia cluster, the autosomal, the RSACs, or in Charlevoix-Saguenay, and then we have SPG7, all the ones I just reviewed. Granted, there are cases everyone. SCA2 patients come from here, but most live in the southern Quebec because that's most of the population. But when they tell you a certain family name and they tell you they have a link to this region, then you know, well, unfortunately, that's what you have. So what has changed from the initial description from Jean-Pierre Bouchard in '78 is that the framework we were thinking of is, well, okay, we have more of those diseases because there's one mutation that's more common. And in fact, most of the cases will have the same mutations. We've learned now that this is really not true. The first statement is partially true. Why we have more of certain ataxias is because one mutation, or at the maximum 2 mutation, are more common in the French Canadians. This being said, mostly the ones that are very large genes like SACs and RSACs, or SIN1 in SCAR8, or the ataxia of both, there's lots of mutations. So many of our patients now are turning out to be a little bit different clinically, have the common founder mutation and another mutation in the gene, and have maybe a little bit of a different presentation, so that we don't pick them up as quickly at the bedside because they have some differences, but then we realize when we do the genetic testing. So we've learned, yes, why we have more in certain regions, and in Quebec in general, is because of 1 or 2 mutations, but we have to be open to the fact that there are more mutations. So even AOA2, we had 7 mutations. We had 5 mutations initially and then more. Same for SPG7, same for SCAR8, and there's more than 10 now. So we have to keep that in mind, because the approach I described, the Charcot approach of saying, well, this patient looks like this patient, well, not always. So we now have founder disease in many conditions in many regions of Quebec. This is already an outdated picture. So we always think of regions, but in every region there's existing or known and others to discover. So just a little bit of background. I won't dwell too much on it because that takes a full lecture, but just to give you an idea of where this comes from. Quebec is a very diverse population to start with, because it was founded mostly by military people and a few sets of women who were orphans and who came from all over France. So we are very diverse. So the deck of card that we received was more limited because it was only about, well, as we'll see in the next card, about 2,200 people contributed to the population before 1680. 1680 is kept as a date because it's the date the last woman arrived. All the rest until we became a British colony, so until there, it was all women that were born in Quebec marrying men coming in. So that's why it creates a kind of heritage that you can dilute, but it's difficult because a lot of the people share those gene pools. Another way to illustrate that is this is a complex slide and I don't want to understand it fully, but it means that if we look at 2 generations, a lot of people are related. If we look at 10 generations French Canadian, most of them are related. So in fact, if you take 2 French Canadians defined as 4 grandparents of French Canadian descent, the likelihood of them sharing one ancestor is 98%. If you go in my region in Saguenay, the likelihood of sharing one ancestor is 100%. The likelihood of sharing 50 ancestors is 100%. So the more you go in regions, the more you will share a gene pool, the more the deck of cards has similarities. And the more the deck of cards has more of certain cards, which unfortunately are associated with disease. Other things that have made this happen as we see it now is Quebec has grown extremely quickly, more than other populations. And compared to France, even during the 19th century, we were growing, the French Canadian were growing at a rate that was 2 to 3 times what was happening in Europe. So that's why it was a tremendous growth for many reasons we can discuss at another time. And behind that was the fact that we had a lot of families, and we always associate with religious behavior. We now know that's not the case. It's just we married early. Families had tradition of marrying early and having large families. And a lot of people did agriculture or on agricultural family or pioneer families where you need a lot of workforce and you need to generate it by having large families and marrying early. And so that's a phenomenon. So we were having, can you imagine in the 17th century, most women were having, on average, were having more than 10 living children. So that's why we're so many, despite the fact that we only descend from about 10,000 people. And of those, only 2,200 are the ones that just are everywhere in our gene pool. So I won't dwell on that. So what we've learned initially, and I think that's when I joined the tradition, the effort to find out about our gene pool, was that if you took Quebec City, which is here, and in fact, this has moved, I don't know why, but if you draw a line just below it, we call this the Northeast Quebec and the Southwest Quebec. And people had come to the conclusion because of OPMD, because of Saguenay disease, that this was the area where we would find all the founder disease because of closer relationship. But in fact, we now know there's some also in the South. So all regions are potential, but there's clearly a lot more in the Northeast, and that has stayed. What has changed dramatically our diagnostic experience, and certainly helping families greatly, is now we don't do single gene testing, despite the fact that we can often predict because of the more limited amounts. We do gene panel sequencing, and that's turning out to be very accurate, obviously, helping us to make diagnosis. So in a clinic, in my ataxia clinic, probably our diagnostic range is over 75% now. And we're finding some of the rare ataxias that we're finding elsewhere in the world. Not everyone has a common ataxia. Some have rare ataxia or rare forms of an ataxia that's more common in Quebec. So we are in another world, and that's having dramatic impact. So let's go to the third part of this talk, which is a change of gear. So you remember we've talked about Bouchard, Jean-Pierre Bouchard, describing this prototypical recessive ataxia. I was not really working on that ataxia. I was working on other ataxias, and then I will thank them later. A foundation asked me, and Jean-Pierre Bouchard asked me, and Jean Mathieu, to try to become more active and try to bring together people to accelerate research on this prototypical disease. It was a time also we were starting to realize that RSACs, as I will call it from now on, was in fact a common ataxia worldwide. So I'll give you a little bit more detail on the disease. I'll talk about the gene and some of the challenges. I'll talk about models and what are we doing to try to correct the phenotype. In other words, finding a treatment. So these are the amazing couple, and I think again it resonates with Jacob's ladder. Sonia Gobeng and Jean Groulot were struck by their first child had RSACs, and they didn't accept that there was little research being done at the time. Unfortunately, since then, their second child has also been diagnosed with the disease, and being very dynamic, brilliant people, knowing a lot of people as lawyers, they decided to develop a foundation oriented on, as we say in French, parce que la recherche guérit, really centered on finding resources, generating researchers, promoting research on RSACs to find a treatment for their children and the others. Very early on, they had a tremendous relationship with the CIHR, the Institute of Genetics, which has been a really full partner on developing this amazing networking effort and collaborative effort. And I can't thank them enough, the foundation and the CIHR. So just to give you an idea, we're now version 3 of networking on having people working on RSACs. We had a first CIHR team grant, and now after that we had a first European team grant called Prepare, and now we have another European team grant, which is called ProSPACs, led by Mathis Snovnik in Tübingen largely, and Rebecca Schula. Can you imagine now we have people from all those countries who have 2 diseases we're proposed to work on in this project. The flagship is RSACs. So we have a European network with a Canadian counterparts, my excellent colleague Cynthia Cagno in Saguenay, and I, and other people. In fact, those are just the PIs. There's at least 45 people in the network. We're working on 2 Ataxias. RSACs being the first, and then another one which is called SPG7, which I mentioned earlier. So that's where we are about a bit over 10 years later, 13 years later, is we have an international network of people. That took time and a lot of encouragement, and clearly the foundation played also a very big role in fostering this interest. So RSAC. So the description of the disease, as I said, was produced in '78. We now know that it's a more variable disease. It goes from in the world. We know that some patients will not learn to walk, will be severely limited intellectually in their development. Some don't even learn to speak. And some will develop RSACs late in their life with only a little bit of gait difficulty. That's a disease. In Quebec, where 90% of the patients have 2 copies of the same mutations, the phenotype is more homogeneous, but the latest work from Cynthia Gagnon has shown very clearly that the phenotype, even with 2 copies of the same mutation, is variable. We have people who walk at 58 with only a cane, and some that stop walking at the age of 17. Same mutation, same 2 mutations. So obviously there's other genes playing a role in this, and certainly we're working on it. But if we go for the way we define it, it's all based on Charles' work. We call it the classical triad. So they have spasticity, pyramidal signs. They have the cerebellar, so the wobbly gait or drunken gait, and with time the drunken voice and the difficulty with the writing. And they have a nerve disease. So in a way, they have a Charcot-Marie-Tooth polyneuropathy associated with it, which also, and the balance between the 3 major system involvement will change the clinical presentation. In Quebec, most people have to go to wheelchair by the age of 41, but it's variable, as I just mentioned. And life expectancy is diminished. It's probably not 51. The more recent data not published, it probably shows that it's later than that, and we have people in their 70s now. There is also a very peculiar retinal change, and that's what we can find on the exam. So in fact, you can make the diagnosis if they have that on the spot. It's specific for this disease. If you have an ataxia and you have those retinal lines, you know it's the disease. And now with time, and in fact, Quebec has contributed, but also the European have developed some radiological criteria. And now the MRI is also pretty specific for this disease. So what do we have pathologically? So then we're going in another, we've talked about the clinic, now we're going to the brain dysfunction. So I think it's appropriate for this lecture where we're talking about neurodegeneration. What we knew is that there were some shrinking of the cerebellum, particularly the vermis, which is really involved in truncal balance. So that had been described. And there was also some loss of the major cerebellar output, which are the Purkinje cells, very, very large cells. Besides that, there was not much known. There was also atrophy of the spinal cord and some loss of myelin. And it's only recently that we, thanks to a discovery that I'll discuss a bit later, we realized that in fact there is a lot of neurofilament bundling, which is all, this is a control, this is an RSAX. You can see this black web is neurofilament bonding. Neurofilaments are important intracellular proteins that are part of the cytoskeleton. So the cell is not jelly, it has also inside a skeleton, a dynamic skeleton, and the neurofilaments, intermediate filaments, are part of the skeleton, which then on it hangs all types of things, including microtubules that are important for transportation of mitochondria and many other vesicles, and so the cell needs a cytoskeleton to bring things generated around the nuclei all the way to the tip of the axons and the dendrites. So one of the reasons why, I must say, I was very not enthusiastic initially to start working on RSAX with this. The gene is huge. It codes for one of the biggest protein in the human proteome, and that's crazy. You're going to work on a protein you don't know the function, a gene where you don't know the function, you don't know really the structure. So that's why we work together as a group, and we've done a lot of work, not only in Canada but elsewhere. And now we know it's a huge protein with specific domains. It interacts with certain important proteins for chaperoning, so it's probably implicated in protein conformation. So we think now this large protein that has a repeated motif helps co-chaperoning. So it's a kind of bench on which large protein are change conformation and then can be probably inserted in the cytoskeleton. And you'll see images why we think this. And so we did a lot of work, including modelization. We are now using this knowledge to develop specific small gene therapies to replace some of its function, like the UBL domain and so forth. So that really we went from 10 years ago having a kind of schematic representation of the protein to now a kind of really functional understanding of the protein, so we can try to use that understanding to develop original treatment. So what we learn is that Saxin is really expressed mostly in the brain. It's not much explained elsewhere. So it's really a neuronal protein expressed in all types of tissues to various degrees. So, and it turns out that where it's expressed the most in the brain is where the disease impacts on the survival with time of these pathways, the cerebellum, some of the tracts, the pyramidal tracts, and some of the cerebellar spinal tracts. So just a few words about function again of Saxin. It's all the idea that it's co-chaperone. A lot of work on that was done by colleagues who were HSP specialists. And we now know that some parts of the protein are really helping to turn around or to help to conform a protein so it can do its proper work. So we're quite convinced that that's one of the defining function. The question is, what does it help fold properly? And that's still the bets are on. We have a few ideas, and many groups have a few ideas, but we don't agree on all of them. But clearly, that will be important to define new treatment modalities. And in that process, more and more data is showing that, in fact, it also helps the normal intracellular dealing with proteins, particularly cytoskeletal proteins that are poorly conformed. So if you remove that, you tend to accumulate many proteins, and there's also a contribution to autophagy that has been developed in the last few years. So this collective work allowed us pretty early in the course to show something that changed a little bit our understanding, and I think will be of interest for the neuroscientist in the virtual room today. One of the things we found initially, in fact, it was Peter McPherson at the Neuro, is that he looked at fibroblasts of patients, and then he looked, this is a mitochondrial, green is a mitochondrial marker. And he realized that the mitochondrial network wasn't the same compared to wild type. So in fact, the mitochondria are deformed, not the right shape. Abundance was not assessed at that time. And then a colleague and collaborator, Paul Chappell in London, produced the first data showing that, in fact, there is a co-localization between mitochondria. Not perfect, but a co-localization between mitochondria and saxin. And then we were able to show that clearly, if you look at a neuron, this is the center of the neuron. Those are neurites coming out of the neuron. And then the red are the mitochondria. You can see how it's beautiful, how the mitochondria are, in fact, moving along those neurites. If you don't have saxin, that's what happens. It clumps around the cell in the cell body, and you see how it just has difficulty going along. It's stuck there. So clearly, that's not good for the cell. And so I think all of us in the world accept that there's a mitochondrial component, slight but truly an important mitochondrial component to cell demise, Purkinje cell demise. And Purkinje cells are peculiar because they're very large cells with a large of dendrites. So it's one of the largest neurons in our body. So it needs a lot of this trafficking. So we now are convinced that the absence of saxin leads to a mitochondrial trafficking. And the question is why? And that's really the two mouse models we developed that really brought us there the most dramatically. So we developed a knockout and a knock-in model that show essentially the same thing. And what it shows is that this is the cerebellum for the ones who are not anatomists, and it should all be brown as it is here. But you see in the mice that some areas are not as brown. That's because there's neuronal loss, in particular Purkinje and dendrites of the Purkinje. So there is loss. You can, if you look at the big Purkinje cell, which is the round red that you see there with their terminal dendrites here, the large dendrites, you can see that normally there's not a lot of neurofilaments in those dendrites. But if you look at the patients, or in fact in the mice, you can see that there's a lot of neurofilament in those dendrites. So the dendritic tree changes in structure. In fact, you can see it very well here also. Look, this is normal mice. Again, the neurofilaments will be shown in brown here. And that's the mice with no saxin. See how all those neurofilaments are there. In fact, it turns out they're not at the right place. They should be in the axon and not clogging the dendritic tree. And that allowed us to go back to the pathology and show that, in fact, if you compare a dendrite in a human to a patient, you have exactly the same phenomena. So yes, we had shown that mitochondria were not the right shape, but then we realized that, in fact, the mitochondria are stuck in an intermediate filament cytoskeleton that's distorted. So, and since then we've published on mitochondrial trafficking and showing that, in fact, it doesn't traffic properly, and maybe even saxin interacts with some of the important proteins that are important for mitochondrial dynamics. We even use that to try to see if we could find drugs, and we use it as a way to assess if a drug could help in accelerated mitochondrial transport. So the mice are one of the, I don't know how many in today on the web talk have had the challenge or lived the challenge of developing mice models. My humble experience has been that 50% they don't help, they don't give a phenotype. But in this case, they gave a dramatic phenotype. Both the knockout and the knock-in gave a very clear phenotype pretty early. At 40 days, we can see that the mice don't walk properly. So now the mice have been used in 2 preclinical trials with really interesting results that may have bearing on future treatment of our patients. One study was done in Italy by Francesca Malteca, and one was done by Alana Watts at McGill. So we are sharing them. They're publicly available. So this is again to show you in more dramatic fashion what happens. You have all these Purkinjes that are so important to control your coordination, and then that's what you have if you have a mutation that interfered with the function of the gene, or if you don't have the gene, you lose them. So naturally, the mice, like the humans, will have a degenerative phenomena and a loss of walking ability and coordination that are unfortunately lead to a severe handicap with time. Interestingly enough, it creates a physiological phenotype in the Purkinje cell, so that also was well documented by our colleague Alana Watt. So now we can also test live cells from these mice, expose them to drugs, and see if those drugs, which can play on channel function, in fact are able to correct some of the abnormalities we see in the sacs. Interestingly enough, sorry. Interestingly enough, those abnormalities are not specific to our sacs. They're very much the same that you see in some of the dominant ataxias. So in fact, the dysfunction, physiological dysfunction, will probably be a universal or largely universal for cerebellar dysfunction in ataxia. So whatever we find that could recalibrate or allow the cells to perform their physiological function may be useful for many of the other ataxia where the cerebellar dysfunction plays a key role in the disability. So we did a lot more work, and I just wanted to show you those images so that they stay in your mind. The patients give the same image as the mice. So mice can be great models. You can see the accumulation of the neurofilaments in the cells. Compare this to normal, and then this to the knock-in or knockout. So we really have an amazing model to do work. And in fact, we as I mentioned earlier, we are looking for, we're going to do more in the next few years, preclinical trials on those mice. And we now share a common protocol that everyone can use to use the mice in the preclinical setting. So what have we learned from these models? We've learned many things. For the mice, both models display an early ataxic phenotype, so great for preclinical trial. Associated with Purkinje loss, so that's also, we have an histological correlate. We talked about the outer firing. They also really much show the accumulation of neurofilament in the same way. So again, if neurofilaments are implicated in other neurodegenerative disorder, that can, whatever we find that will change the dynamics of neurofilament in the neuron may have relevance for other disease. And what's really of great use, and I'll show you in the next 2 slides, is that we realized early on that we can see cytoskeletal or intermediate filament bundling in other cells. Look, these are human fibroblasts. That's the normal. I showed you earlier the mitochondria. This is not the, this is the vimantin, which neurofilaments are not expressed in fibroblasts. So vimantin is the major intermediate filament. It looks great. It's and really aesthetic. And then you go to the sacs. So the patients, or in fact, this is a knockout, a CRISPR knockout. You see this abnormal network of intermediate filaments and what we call bundling, this kind of clumps. So you can see that in HEC cells. If you knock out the gene, you can see that in neuroblastoma-derived cells, and you can see them in cultures from the mice that can also be used to test drugs. So it's a universal thing. So whatever we find may help a lot of, we can find using all those cell lines. So that's what we So though we don't know exactly where the the intermediate filament bundling, where does it cause the problem? Does it really in the somatic, so the neuronal cell body, or is it more in the axon, or is it more in the dendrite? I think we know there's pathology everywhere, including the axon. So at this point, the search is to find out just simple ways to make this cytoskeleton better. At the same time, trying to find what tips the cell from a functional to a dysfunctional physiologically, and then from a physiologically partially incompetent cell to death. Those two things. And I can't tell you now because the story is not out as to what's the key thing for the dysfunction and what's the key thing for cell death. But clearly that will help us understand this disease and probably many others. And so if we go to the idea of using those bundling for screening, so these are the neurofilament. This is the fibroblast, immortalized fibroblast, and we've developed with the great team of Eric in the Institut de Recherche en Immunologie and Cancérologie of Université de Montréal, directed by Michel Bouvier and his high throughput platform. We developed a very robust assay to use to screen molecules, and that has been an adventure by itself. I didn't think that we would be there, and so at this point we screen over 25,000 molecules, including proprietary libraries from Pfizer and Takeda, and they are with us to some extent, but largely it's their work concentrating on a new series, a new family of medication we found that in fact allow control cells. There's no impact of the drug, but if you go from patient cells that have bundling, almost 100% resolution of the bundling within 24 hours with no cell division. So that's what we're using now to try to take this new drug series to try to make it a drug that we could expose the mice to. And I think we're very close to that, hopefully in the next few months. And at the same time, trying to see if we can develop partnerships to bring this new drug to treatment, while at the same time using this model to validate other hits from other groups. So it's a very exciting time for research. So yes, we're 13 years later. It takes too much time, but we're really testing drugs in mice and finding new drugs. So I think we're getting there. So all this work would not have been possible without the help of many people. But I need to thank again Helen and Jeff Schwartz for this wonderful award, which I think will bring a lot of visibility to the work on RSACs and ataxia in Quebec. The foundations that help us all the way, as I said, the Institute of Genetics, in particular muscular dystrophy, that has always played a role in promoting research on RSACs for over 20 years in Quebec. Our two networks are European networks, European funded networks of Prepare, which is now passed on to a new version of ProSpacs, and a very generous family in Montreal, the Monaco Foundation, that have supported ataxia research in my lab for close to 10 years now, and has made a big difference for many of the projects I've discussed. For the ataxia project, I wanted to thank Alexandre Rivière, who unfortunately has left my lab now, who did a lot of all of the mouse work. Marie-Josée Dicker, who's my eternal accomplice, who's been working with me since the beginning of the lab, and my wonderful secretary coordinator, Catherine Boyer, who has this magic touch with patients. One of the challenges of a clinician scientist is to keep the right balance between helping patients and having patients help me to make projects go forward. And in the middle of that is that relationship where there's no confusion as to those two parts of the relationship. And Catherine has been wonderful in ensuring that I stay a very patient-centric physician and not a project-centric physician. So I thank them both, and numerous students over the years. Some of them have their independent career and have stayed in the ataxia field or other fields will develop. Wonderful colleagues at McGill that have helped me to develop. Eric Shoebridge, who has been a support. He's the co-director of my unit now, and really important person for much of our work. Benoit Coulombe, who helps us with some of the proteomics work, the tremendous team at the IRIC, and I would underline François Lavallée, who's the medical chemist who's done a lot of the modification, and Karine Audette and Jean Duchesne have been amazing at the drug screening exercise, and Manon has helped us, and Marnier, who's helping us to move it to another stage. And clearly my mentors, friends, this is a tradition. Neurogenetics is very neuromuscular in Quebec, and I thank them again. And my younger colleague, Cynthia Gagnon, who's been a partner in crime in all the Ataxia field and has been playing a major leadership role now internationally in this field, and my other colleagues elsewhere who have joined at different times the RSAC's international group effort. So on that, I thank you for your patience, and thank you again for the prize, Ellen and Jeff, and I'm looking forward to answering your question. Thank you.
Thank you very much, Bernard, and for a wonderful lecture. We have several questions, so I would like to start answering some of these questions. One is, Dr. Dowling says, "Hi Bernard, great talk. The failed trafficking pathology, particularly of neurofilaments, reminds me of what is seen with giga-axonin knockout and BPAG1 dystonin knockouts. Does Saxon directly interact or stabilize these proteins, or is this just an example of shared pathology from disturbing a common pathway?"
So there's no clear relationship that I know of, but the idea has been taken up. So we have a young colleague here, Benoit Gentil, who is doing a shared project where he studies both disease and tries to see the interface between the two. So yes, the hypothesis that maybe there is a relationship between the two is being explored, but a direct relationship between the two proteins has not been established to my knowledge.
So another question comes from Grace Yoon. "Thanks for the wonderful presentation. Why are Purkinje cells so sensitive to SAX deficiency compared to other cerebellar cell types?"
Thank you, Grace, for the great question. So I think the kind of textbook answer to that is, as I mentioned earlier, Purkinje cells are huge, but they also have the largest neuritic arborization. The dendritic tree is huge. It's like I have no hair, but imagine me as the soma of the Purkinje cell and put me an afro wig on. That's what a Purkinje looks like. Most neurons would have just a normal haircut like Jeff Schwartz, but it's so different. So the problem is that requires a lot of contacts with many cells. That's why the arborization is so large. A lot of additive physiology of signal, and it requires a lot of energy and a lot of transportation. So it's a cell that's really high functioning, integrative neuron that just needs all this contact, and therefore is a high energy, highly calcium and mitochondrial dependent neuron. So I think all of those are why we're finding it. In fact, the axonal pathology, which we see in other diseases, is not as so striking in RSACs. There is a little bit of axonal pathology, and there is a little bit of axonal terminus pathology, but it's not the most dramatic. It's really the dendritic pathology.
That was a great answer. Jihei Park is saying so much for the wonderful talk. I have learned a lot about the history and genetics of ataxia. The Saxon project was very interesting, especially the increased neurofilament levels in the knockout mice. Do you know how loss of Saxon leads to neurofilament accumulation?
So the short answer is we don't know how. What we know, however, is that Saxon interacts directly with the neurofilament. So as I described, Saxon being a kind of workbench and a chaperone, so it has arms that are probably helping to conform proteins. Some of those proteins are the neurofilaments. That's the hypothesis. So the idea is if you lose the protein's not there, the neurofilaments are produced, and they're looking for the right conformation, the right way to set, and they're missing this bench. And so they're they're not doing it properly. They're not being phosphorylated properly. And therefore, not being phosphorylated properly, they don't go in the right compartment of the cell. So they're sent to the dendrite rather than going to the axon where they should go. So it's a set of abnormalities that are happening. So we have insight on some of those, but clearly, I think it's very clear now that the neurofilaments have a direct interaction with saxin.
Okay, thank you. I think we're running a bit behind, so I'm going to end with some questions about the genetics. We'll switch from saxin to the genetics. So these questions come from Joanna Romans. And I had a couple of these same questions. So in retrospect, were the array of variants found in Quebec for an individual condition, as in more than one or two per disease, an indication that some of the more common ataxias should perhaps not be so rare in other places?
So I would frame it differently. So the idea is, what we've learned is, yes, we're better at describing, in a sense, and then that's also the talent of Jean-Pierre Bouchard. But we enter the museum and there's just less statues to look at. And so, yes, but the reality, it's still a museum of the history of humanity. And the major classic statues are in all the museums. It's all the same Greek statue everywhere. And so, yes, what we find at home will be also present elsewhere. And I think what the low-lying fruit for us, and I don't like the expression particularly, but I will use it again tomorrow, but the low-lying fruit were the common looking the same. And that was common looking the same is because it was the product of those more common mutations. But the common was common elsewhere, not to the same degree. I mean, absolutely not. I mean, we have a real health challenge because some of them are really common in our population. I don't know if it answers, but the question, which is a hard one to answer fully.
Yeah. Joanna also wants to say it's really nice to see you. And are the multiple variants per disease just a reflection of initial population, or is there another?
So, sorry, that's a good question. It fascinates me as a historian, and I need to go back to that. We did a lot of, Babo introduced really the use of genealogies to try to, because Quebec has all the genealogy now in an informatic way, thought that we could use that to understand the introduction of mutations. I think at this point, except for one disease I worked with Eric Shoebridge, which is Leon, so it's Leber's optic atrophy, I don't think we have completely convincing evidence that we introduce only who introduced the mutation and whether there was a single introduction in the population. And I would even favor some of the mutation may not be European, they may be First Nation mutations that were introduced in the population. We just don't know. So there's much work to be done to better understand number of mutations, who introduced them, what's the impact. And I wouldn't be surprised that some of the mutations were already in North America before the arrival of the European, though OPMD probably not, certainly Leber optic atrophy not, but the others we'll have to see. It's still up for grabs. I mean, there's a lot of excellent demographic genetics in Quebec being done, and the models are so complicated that even I lose my French.
Okay, I'm going to ask one more question, and this is from Ralph Fullman, because it's related to this. Sociologically, over the many years, did the affected population realize the inheritance pattern of these diseases and try to avoid cousin intermarriage? And does CRISPR technology have any role in therapy?
So I usually always mention that. I didn't this time because of time, but so someone using genealogies and marriage records that were available all over the world, looked at which population in the world marries the least between first and second degree cousins. And it turns out it's the French Canadians. We don't marry between first and second degree cousins, though there are some, but it's rare. Because of a religious taboo and a religious cost, it was not done. So we're just very close knit, but not practicing first and second degree cousin marriage. And so that's kind of striking, but that's a reality. So it's really sharing some of cards because of shared history rather than this happening. This being said, we do, like in every other population, have close inter-cousin marriage, and we do have some cases, and some of the projects, these couples made the big difference for us to find the mutation. As to CRISPR treatment, there's many labs in Quebec who are trying to use CRISPR to treat or design treatments for some of the common, my lab's not involved, but some of the common mutations. So yes, this is being very actively explored by some.
Okay, I would like to draw this to a close and thank Dr. Brais for a wonderful presentation. And we will move on to the award ceremony at this point. So to present the award, I'd first like to introduce our CEO of Sick Kids, Dr. Ronald Cohen, who has agreed to say a few.
Good morning, everyone. It's my pleasure actually to be here, and thank you for joining us today, celebrating the Norman Saunders Jacob's Ladder International Research Prize Lecture. Let me first start by saying that Jacob and his absolutely wonderful family have been an inspiration for so many of us here at Sick Kids for years and years, but also really for thousands of individuals outside of Sick Kids. I think it's fair to say that Jacob and his family have touched so many lives, and you continue to do so. I would like to thank you, Ellen and Jeff, wholeheartedly for your ongoing generous support that has, on one hand, allowed us to perform groundbreaking research in the area of neurodegenerative diseases, which we really could not have done without you, but it has also allowed us to really think about the care of children with complex medical conditions in a completely new and different way, and really has facilitated the establishment of the Norman Saunders Complex Care Program here at Sick Kids. And I could really not think about a better way of honoring Dr. Saunders in association with Jacob's Ladder, because like Jacob, Dr. Saunders has been an inspiration for generations of pediatricians, for their patients and families. I think it's very fair to say that Dr. Saunders was the pediatricians of all pediatricians. And there have been numerous times when I spoke to people about him since I arrived here, when they say, I just want to be like him. And I remember once reading an article from Ian Brown, I think it was in 2006, in the Globe and Mail, where he called Dr. Saunders the hero of our city. And obviously, Dr. Saunders was also Jacob's pediatrician. And in that same article, I remember reading how Ellen commented on the fact that Dr. Saunders was visiting Jacob on a daily basis whenever he had to be in the hospital. And I think that just in a small part speaks volume to who he is. And it's really because of people like him, Jacob, Ellen, and Jeff that our world is in a better place, no matter what the outside circumstances are. So Ellen and Jeff, thank you wholeheartedly for your strong and ongoing support. The legacy that Jacob has created will live here forever, but not just within, but also outside of the walls of Sick Kids. So thank you very much for everything that you have been and continue to do.
Thank you, Dr. Cohen. That was a very warm welcome and introduction, and thank you all for being here, especially Dr. Brais. It was a fantastic presentation. Appreciate that. I just want to say you've really talked about Norm in the way that we thought about Norm, and he is the namesake of this award because he was the brainchild behind this award. And truth be told, if it were up to me, we probably would be one of those families that kind of retreated into the shadows and dealt with this as a family internally, but my lovely wife said, no, that's not the way we're going to do things. And we're going to bring value and benefit to the community from what we've learned. And we're going to share all the benefits of having a child with special needs. And she was right, absolutely right. And I think Norm concurred with her on that. And as not only our pediatrician, but a member of our board, and a guiding light for us in many different ways, he said, you know, you have to share this. You have to be able to be open about it. And he came up with this idea for the award, which we have been doing for years and have an unbelievable roster of awardees across the globe. And he said, this is an opportunity not just for you to share your story, but also to share with people, to have people who win the award share their findings with the people here in Toronto and beyond, which has ultimately resulted in many different collaborations. And I think that's what it's all about. I think we can't lose sight of that, especially in light of the pandemic, that we have to be able to share information so that we can move these projects forward at a quicker pace. So absolutely thrilled to be able to continue this. I want to thank the committee for selecting Dr. Brais and people going forward and people going back. They've done an unbelievable job. It's a volunteer role for them. And I think they've really hit the nail on the head every time they've selected an awardee. So we're truly appreciative of that, as it reflects the ideals of both Ellen and myself, but in addition, the award itself. Now, before I get to the presentation, and thank you for putting that slide up, we closed Jacob's Ladder at the end of 2019. But what was really important to us is that we wanted to make sure this international award continued on for many years. And we were in the position to actually set up, with the help of the foundation at Sick Kids, an endowment fund that would allow this to go on almost 20 plus years from now, based on the funding. So we're thrilled to be able to do that and announce that, because it was one of the most important things that we as a foundation did. So without further ado, I want to thank Dr. Brais for his presentation, and in conversation with him over the last little while, he couldn't be a better recipient for what we're going through now and what we anticipate will benefit genetics and neurogenetics into the future. So thank you very much, Dr. Brais.
Very welcome. Thank you. So thank you for, I believe I have to say a few words, and thank you very much. I'm honored, and to me, it's a great moment to salute parents who decide to do two things: take care of their child the best possible way, and share with others what they've learned through it, and doing the best they can for their parents. They realize, I think, talking to physicians, Saunders and others, that it means promoting research. Research is hope, but it's hope that, I hope I described that in the RSAC section. It's hope that diagnosis leads one day to treatment. So the experience of the parent with their child that's changing and not going through very difficult time will not happen again. And so that, I think, Jacob's Ladder and this awardee is to do that, is to ensure that we bring hope and show that we're not forgetting Jacob and not forgetting you as parents, that we will work hard to find ways to treat those children that are still being born, and they will always be born. And we need to never forget them. So I think this award is a way to never forget. I thank you again.
Thank you so much, Bernard. That was a wonderful way to put it. I would like to also thank the Schwartz family for supporting this endowed lectureship. And this has occurred over the last 20 years already. And thank you so much for donating enough money to ensure that it keeps going for the next 20 years. Now, I'd like to introduce Ellen Schwartz, who is Jacob's mother. and who has documented many of Jacob's lessons about life through writing books. But also she's going to tell you a bit about Jacob's love of life and of music.
Hi, everyone. Thank you so much. Congratulations, Dr. Brais, for being an honorary winner of this award that we are all so very proud of, because we just love and we are so dedicated and grateful to Sick Kids and all they've done for us. And having Ronnie and Monica and Norm Saunders and Jeremy Friedman and their whole cast of team. And they've just been so loving and so supportive. And it's our honor to always, for the next 20 years, carry on this legacy. So what an honor. Jake, as many of you know, wasn't able to walk or talk or see. And, you know, we were told that he probably wouldn't live to his 10th birthday. Well, miraculously enough, he lived till 21. And he taught us so much about life and living that he even made me a writer, and I'm not a writer, but I had so many lessons to share from him. So I did. And one thing that Jacob loved was music. He was able to communicate through music. He just lit up when he heard it. And because of that, so many talented young people used to come to our house on Sundays to sing to him. And on his 20th birthday, we got them all together and we said, guys, let's write a song. And we wrote this song, and the words are, if Jacob could speak, these are the words he would say. So I'm going to let you see that song and see pictures of Jakey, and I hope you enjoy it. It's called I Rise Above. Thanks.
Coming from the cold on a Sunday, I've been waiting for you. Pick up your guitar and play me a song. Let the music lift our souls. I love the sound of your voice and your fingers on the strings. This moment
Wow. Thank you so much. And what a group of budding musicians. I swear that was wonderful. I want to thank Bernard, Ronnie, the Schwartzes, everyone for their participation today. This has been a very special Jacob's Ladder presentation, remotely due to our circumstances, but thank you all, and we'll draw this to a close.
you.
Remember that Bernard also gives genetics rounds tomorrow morning at 9. Thank you. Bye.